Today, I want to have a discussion on cell organelles. So, what are cell organelles? So, the cellular components, cell organelles are cellular components. So, these organelles include both membrane and non-membrane bound organelles, which are present within cells and are distinct in their structures and functions. So these cell organelles coordinate and function efficiently for the normal functioning of the cell. So a few of them function by providing shape and support, whereas others are involved in the locomotion and reproduction of a cell. So there are various organelles present within the cell and are classified into three categories, and that is based on the presence or absence of membrane. So organelles without membrane. So these are the cell wall, ribosomes, and cytoskeleton are non-membrane bound cell organelles. So these ones are present both in the prokaryotic cell and the eukaryotic cell. Then we move on to the single membrane bound organelles. These are the vacuole, lysosome, Golgi apparatus, endoplasmic reticulum, and these are the single membrane bound organelles are present only in a eukaryotic cell. Then we have the third category, which is the double membrane bound organelles, and these are the nucleus, mitochondria, and chloroplast, which are double membrane bound organelles present only in a eukaryotic cell. So, what are the functions of these organelles? So, I would like us to start with plasma membrane. So, the plasma membrane is also termed as a cell membrane or cytoplasmic membrane. So, it is a selectively permeable membrane of the cells, which is composed of a lipid bilayer and proteins. So, the plasma membrane is present both in plant and animal cells. So, it functions as the selectively permeable membrane, that is, it permits the entry of selective materials in and out of the cell according to the requirement. In an animal cell, the cell membrane functions by providing shape and protects the inner contents of the cell. So, based on the structure of the plasma membrane, it is regarded as the fluid mosaic model. So, according to the fluid mosaic model, the plasma membranes are subcellular structures made of a lipid bilayer in which the protein molecules are embedded. Then we move on to the cytoplasm. So, what is the cytoplasm? So, the cytoplasm is present both in plants and animal cells. So, these are jelly like substances found between the cell membrane and the nucleus. So, they are mainly composed of water, organic and inorganic compounds. So, the cytoplasm is one of the essential components of the cell where all the cell organelles are embedded. So these cell organelles contain enzymes mainly responsible for controlling all the metabolic activities which are taking place within the cell and are the site for most of the chemical reactions within the cell. The third is the nucleus. So the nucleus is a double membrane organelle and it is found in all eukaryotic cells. It is the largest organelle which functions as the control center of the cellular activities and is the storehouse of the cell's DNA. So by structure, the nucleus is dark, round, surrounded by a nuclear membrane. It is a porous membrane and forms a wall between cytoplasm and nucleus. So within the nucleus, there are tiny spherical bodies called nucleolus. So it also carries an essential structure called the chromosomes. So what are chromosomes? Chromosomes are thin and thread-like structures which carry another important structure called a gene. So genes are hereditary units in an organism. For example, they help the inheritance of traits from one generation to another. Hence, The function of the nucleus is to monitor cellular activities, including metabolism and growth by making use of DNA's genetic information. So nucleoli in the nucleus are responsible for the synthesis of protein and RNA. And then the endoplasmic reticulum. So the endoplasmic reticulum is a network of membranous canals filled with fluid. So they are the transport system of the cell involved in transporting materials throughout the cell. So there are two different types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So what are the functions of the endoplasmic reticulum? So the rough endoplasmic reticulum, these are composed of cisternae, tubules, and vesicles, which are found throughout the cell and they are involved in protein manufacture. What about the smooth endoplasmic reticulum? So they are the storage of organelles associated with the production of lipids, steroids, and are also responsible for detoxifying the cell. Then we move on to the powerhouse of the cell, that is the mitochondria. So mitochondria are called the powerhouses of the cell as they produce energy-rich molecules for the cell. So the mitochondrial genome is inherited maternally in several organisms. So what is the mitochondria? The mitochondria is a it's a double membrane bound sausage-shaped organelle found in almost all eukaryotic cells. So the double membranes divide its lumen into two distinct aqueous compartments. That is the inner compartment, which is called a matrix, which is folded into cristae, whereas the outer membrane forms a continuous boundary with the cytoplasm. So they usually vary in their size and are found either round or oval in shape. So mitochondria are the sites of aerobic respiration in the cell. They produce energy in the form of ATP and help in the transformation of the molecule. For instance, glucose is converted into adenosine triphosphate, that is ATP. So mitochondria have their own circular DNA, RNA molecules, ribosomes, and a few other molecules that help in protein synthesis. Then we move on to plastid. So plastids are large membrane-bound organelles which contain pigments. So based on the type of pigments, plastids are of three different types. So we have the chloroplast, chromoplast, and the leucoplast. So what is the function of the chloroplast? So chloroplasts are double membrane-bound organelles which usually vary in their shape from a disc shape to spherical, discoid, oval, and ribbon. So they are present in mesophyll cells of leaves, which store chloroplast and other carotenoid pigments. So these pigments are responsible for trapping light, energy, which is used for photosynthesis. So the inner membrane encloses a space called the stroma. So flattened disc-like chlorophyll-containing structures, known as the thylakoids, are arranged in a stacked manner like a pile of coins. So each pile is called a granum, and the thylakoids of different grana are connected by flat membranous tubes known as stromolamella. So we move on to the chromoplasts. So the chromoplasts are So the chromoplasts include fat-soluble carotenoid pigments like xanthophylls and carotene and others. And these ones are going to provide the plant with their characteristics, color, that is yellow, orange. Yeah. So then we move on to leucoplasts. So leucoplasts are colorless plastids which store nutrients. So amyloplasts store carbohydrates, aleuroplasts store proteins, and elyroplasts store oils and fats. Then we move on to the ribosomes. So ribosomes are non-membrane bound and are important cytoplasmic organelles, and they are found in close association with the endoplasmic reticulum. So ribosomes are found in the form of tiny particles in large number of cells and are mainly composed of two-thirds of RNA and a third of proteins. So they are named as the 70s found in the 70s found in prokaryotes, or the 80s found in eukaryotes. Okay, so ribosomes are either encompassed within the endoplasmic reticulum or are freely traced in the cell's cytoplasm. So ribosomal RNA and ribosomal proteins are the two components that together constitute the ribosomes. So the primary function of the ribosomes include protein synthesis in all living cells that ensure the survival of the cells. Then we move to the Golgi apparatus. So the Golgi apparatus is also termed as the Golgi complex. It is a membrane-bound organelle which is mainly composed of a series of flattened, stacked pouches called the cisternae. So this cell organelle is primarily responsible for transporting, modifying, and packaging proteins and lipids to targeted destinations. So Golgi apparatus is found within the cytoplasm of a cell and is present in both plants and animal cells. Then we have the microbodies. What are the microbodies? So microbodies are membrane-bound, minute, vesicular organelles found in both plants and animal cells. So these ones contain various enzymes and proteins and can be visualized only under the electron microscope. Then the cytoskeleton. So the cytoskeleton is a continuous network of filamentous proteinaceous structures that run throughout the cytoplasm. So from the nucleus to the plasma membrane, and it is found in all living cells, notably in the eukaryotes. So the cytoskeleton matrix is composed of different types of proteins that can divide rapidly or disassemble depending on the requirements of the cells. So the primary functions include providing the shape and mechanical resistance to the cell against deformation. The contractile nature of the filaments also help in motility during cytokinesis. Then we move on to cilia and flagella. So cilia are hair-like projection, small structures, and these ones are found outside the cell and work like to either move the cell or the extracellular fluid. So flagella are slightly bigger and are responsible for the cell movements. So the eukaryotic flagellum structurally differs from its prokaryotic counterpart. So the core of the cilium and flagellum is called an axonym, and this one contains 9 pairs of gradually arranged peripheral microtubules and are a set of central microtubules running parallel to the axis. So the central tubules are interconnected by a bridge and are appended by a central sheath. So one of the peripheral microtubular pairs is also interconnected to the central sheath by a radial spoke. Hence, we have a total of 9 radial spokes. So the cilia and flagella emerge from central-like structures called the basal bodies. So we move on to centrosome and the centrioles. So the centrosome organelle is made up of two mutually perpendicular structures known as the centrioles. So each centriole is composed of 9 equally spaced peripheral fibrils of tubulin proteins, and the fibril is a set of interlinked triplets. So the centrioles from the basal bodies of the cilia and flagella also they are going to give rise to spindle fibers during cell division. Then we move to the vacuoles. So vacuoles are mostly defined as they are the storage bubbles of irregular shapes which are found in cells. They are fluid-filled organelles enclosed by a membrane. So the vacuoles stores the food or a variety of nutrients that a cell might need to survive. So in addition to this, it also stores waste product. So the waste products are eventually thrown out by vacuoles. Thus, the cell is protected from contamination. So the animal and plant cells have different size and number of vacuoles compared to the animals, plants, and they also have uh, larger vacuoles. Okay. So with this, I would like to ask you a question. What would you like us to, or would you like me to discuss in next video? And let me know your comment if there is something you need to be improved about these little tutorials. And thank you for your time.